In this video, we can discuss about the microbiology of fungi. In this, we will discuss the definition, different properties, different classification, morphological and taxonomical classification, reproduction, cultivation and applications of fungi. Let's start with the introduction of fungi. Microbiologists use the term fungus to include non-motile, eukaryotic, obligative or facultative aerobes, spore-bearing organisms with absorptive nutrition which are reproduced sexually or asexually. The term mycology is referred to the study of fungi, and mycotoxicology is the study of fungal toxin and their effects, and mycosis is the study of disease caused by fungi in animals. Now coming to the distribution of fungi, where the fungi can be observed. It is widely distributed and are found in dark places where low moisture content is present, usually grown better in an environment with pH of 5 and which require less nitrogen than the bacteria, which are abundant in soil and which are seen on vegetative and in bodies of water where they live largely on decaying leaves of wood. Primarily, these are terrestrial organisms, although few are seen in freshwater or marine. Fungi can be seen in beneficial relationship with other organisms like mycorrhiza in association with roots of vascular plants, lichen in association with algae. When we are comparing the fungus with other microorganisms, few are pathogenic and they will infect the plants and animals. For example, out of 10,000, only 100 will be pathogenic to human and animals. Now coming to the nutrition of fungi. Nutrition of fungi is relatively simple and which show wide range of growth rate. Most of the fungi are saprophytes. What is meant by saprophytes? Which depends on the dead organic matter. So this fungi will produce various hydrolytic enzyme which is known as exoenzyme, and so that which will degrade complex insoluble organic matter into soluble simple organic and inorganic molecules, and these molecules are majorly absorbed by the fungi through their plasma membrane than ingesting them. So these are scavengers and which will act as an promoter of soil fertilizers. So they will form an integral part of nitrogen, carbon and phosphorus cycle. So that is the nutrition of fungi. Now coming to other properties. Fungi cells are enclosed in cell wall of chitin, which is an polysaccharide which is composed of N-acetyl glucosamine, which we had discussed in the structure of gram-positive bacterial peptidoglycan layer. And most of the fungi are aerobic except some yeast which is an facultative anaerobe and rumen of calta which is an obligative anaerobes. All the fungi are non-flagellated and non-motile, and they carry a vegetative structure which is known as thallus, and the structure of thallus may vary in their complexity as well as the size range from single cell microscopic like in yeast to the multicellular mold macroscopic filamentous colonies like in off-balls or mushrooms. And there are two types of reproductive structures in the fungi: sporangia from asexual spores and gametangia from sexual gametes. Now coming to the morphological classification or structure of fungi. Morphologically, fungi is classified into yeast-like fungus, mold or filamentous or mycelial fungi, and dimorphic fungi. We can discuss each in detail. First one is yeast. These are unicellular, non-filamentous, oval or spherical shaped fungus, which is usually a size range of 3 micrometer in diameter, and which are widely distributed in nature and which are found as white powdery coating on fruits and leaves. They will reproduce either asexually by budding or by transverse division. Cell wall may consist 2 to 3 layer of chitin. Within the cytoplasm, a well differentiated nucleus, numerous vacuoles containing food and waste substances, and granules of glycogens or protein may be present. This yeast can be cultured in SABOURAUD'S agar media for creamy mucoid colonies. The only pathogenic yeast of medical importance, Cryptococcus neoformans. So this is the structure. This is a unicellular, ovoidal or spherical structured fungus, that is the yeast. Now coming to the second morphological classification, thus that is yeast-like fungus. They grow partly as yeast and partly as chain of elongated budding cells which are joined end to end forming an pseudomycelia. These are separate cell but they are joined by end to end to form like an pseudomycelia as in the advanced fungus. So a perfect example is Candida, in which the bud remain attached to the mother cell and elongated, followed by repeated budding forming chains of elongated cell, which is known as pseudohyphae. So that is the yeast-like fungus. Now coming to the third morphological classification, that is mold or filamentous or mycelial fungi. Fungus grows as branching filamentous, which is known as hyphae. This is the principal element of growing of vegetative form of mold, and these hyphae may continue to grow and branch to form tangled masses of growth called mycelium. And sometimes in the molds, the hyphae contain cross wall called septae, which will divide them into distinct uninucleated cell-like units, and this type of hyphae is known as septate hyphae. And in few classes there will not be any septa, and which will appear as long and continuous cell with many nucleus, which is known as cenocytic hyphae or aseptate hyphae. So this is an septate hyphae in which it is divided into separate uninucleated cells. Here this is cenocytic or aseptate hyphae, which is presented as long tube like structures without any septa. Now these hyphae are composed of outer cell wall and an inner lumen which contain cystoles, organelles. A plasma membrane surround the cytoplasm and lie next to the cell wall. The portion of hyphae that obtain nutrient is known as vegetative hyphae, and the portion of hyphae which is concerned with the reproduction is known as reproductive or aerial hyphae, which often bear some reproductive spores. The hyphae grow by elongation at their tips, which is known as apical growth, or by producing side branches. Now coming to the last morphological classification of fungus, that is dimorphic fungus. These are the fungi which will exist either as yeast or as mold depending on the growth conditions. The yeast forms are called parasitic form, which occur in host tissue and in an in vitro condition at 37 degrees Celsius, or it may occur as filamentous form, which is known as saprophytic phase, which occur at soil at 20 to 25 degrees Celsius. Most of the fungi causing systemic infection belong to this classification, like Histoplasma capsulatum, Blastomyces denitidis. These are the dimorphic fungus. Now coming to the next classification, that is taxonomical classification of fungi. Fungi are placed in the classification of phylum Thallophyta on the basis of formation of sexual spores. They have been divided into four distinct classes: zygomycetes, ascomycetes, basidiomycetes, and eumycetes or fungi imperfecta. Now coming to the zygomycetes, these include lower fungi with non-septate hyphae and which will form asexual spore of sporangiospores containing within the sac-like structure called sporangia, and also it will form sexual spore, oospores and zygospores. This is a structure of an zygomycetes with an swollen structure on the tip of the elongated tube, that is the zygomycetes. Second one is ascomycetes. They possess septate hyphae and form sexual spores called ascospore in sac called ascus. Now coming to the third classification, that is basidiomycetes. They also possess septate hyphae and also form sexual spores called basidiophore outside on the tip of pedestal called basidium. And last one is deuteromycetes, fungi imperfecta. They also possess septate hyphae but do not produce sexual spores. Most of the fungi of medical importance belong to this class. Now we can discuss the reproduction of fungi. Reproduction of fungi can be either asexual or sexual. First one is asexual reproduction, which can be accomplished by several ways. First method is a parent cell can divide into two daughter cells by constriction and formation of a new cell wall. And second method is somatic vegetative cell may bed to produce new organism. This is the most common method in yeast. And third one is, and the most common method of asexual reproduction is spore production. Asexual spore formation occur in individual fungus through mitosis and subsequent cell division. There are several types of asexual spores. So coming to asexual reproduction by budding, in this parent cell will form an protuberance which is known as bud on its outer surface, and as the bud elongate and the parent cell nucleus divide and the on nucleus will migrate into the bud, the cell wall materials is then lie down between the bud and parent cells, and then bud are eventually break away. The budding is the most common and most distinctive method of asexual reproduction in the fungus. Now coming to the asexual reproduction by asexual spores, these are the product of mitosis which will form in an sporangiospore. These are the spore inside a swollen fertile structure called sporangia. There are different type of asexual spore, canidiospores and thallospore. First one is calidiospore. If the spores are not enclosed in a sac but supported by a fertile structure, it is known as canidiospore. And second one is thallospore. In this, spore resulting from change in the vegetative hyphae or thallus, and which can be divided again into three. First one is arthrospore. In this, hyphae fragmenting into small pore with a thickened cell wall. And second one is chlamydiospore. In this, hyphae is divided into spore-like cell with large food reserve and resistant to the unfavorable environment. And third one is blastospores. In this, spore produced from the vegetative mother cell by budding. So these are the different asexual spore which are used for the asexual reproduction of fungi. So these are the different structures of asexual spore. This is the sporangia, which is seen as an sac in the top of the pedal, and this is the canidiospores. Then this is arthrospores, this is chlamydiospores, and this is blastospores. Now coming to the sexual reproduction of fungi. Usually the sexual reproduction involve the union of compatible nucleus. Some fungal species are self-fertilizing and will produce sexually compatible gamete on same mycelia, which is known as homothallic. In other species, which require outcrossing between different but sexually compatible mycelia, which is known as heterothallic. Depending on the species, sexual fusion may occur between the haploid gametes. These gametes produce bodies called gametangia or hyphae. Sometimes both cytoplasm and haploid nucleus fuse immediately to produce the diploid zygotes. So these are the different sexual spore which are the product of meiosis. First one is ascospores. These are the spores produced in sac or ascus. Second one is basidiospores. These are the spores produced at the surface of club shaped structure, that is the basidium. And next one is zygospore. These are the spores produced by the fusion of similar appearing gametes formed at the tip of hyphae. And last one is oospore. These are the spores resulting from the mating of two unlike gametes. So these are the different sexual spore which are used for the sexual reproduction of fungus. Now coming to the cultivation of fungus. Fungus usually grow better in an environment with pH of 5 and which require less nitrogen than the bacteria when compared with other organisms. Fungi usually absorb nutrients and they do not ingest them. They are heterotrophs. The absorption is aided by the enzymes secreted in the environment, which will break down the large organic molecules into smaller ones, and this can be directly passed into the cell. For the requirement of fungal growth, we have to include sugar, inorganic and organic nitrogen sources, minerals, vitamins, and sometimes need complex media with peptone and meat extract. Media for fungal growth have a higher sugar concentration, low pH between 3.8 and 5 than the media for the bacterial growth. So this is a common media which is used for the cultivation of fungi, that is known as SABOURAUD'S agar media, which consists of peptone, glucose, water, agar, and peptone of 10 gram and glucose 40 gram and agar 15 gram for 1000 ml, and we have to adjust the pH of SABOURAUD'S agar media to 5.6. So that is the cultivation of fungi. Now these are the some diseases which are caused by fungus. These are the some pathogenic fungi. Blastomyces dermatitis, which will cause blastomycosis. Candida albicans, which will cause candidiasis. Histoplasm capsulata, histoplasmosis. So these are some examples of disease which are caused by different pathogenic fungi. Now coming to the most important topic of fungus, that is the application of fungi. When we are comparing the application of fungi with other microorganisms, fungi are the most commonly used organism for the industrial scale production of different pharmaceutical products. Fungi have different pharmaceutical or industrial application. First one is for the production of non-antibiotic specialty compounds like polyethers. An example for polyethers is monensis, and it is obtained from Streptomyces cinnamonasis, which is an anti-protozoal agent. And second one is ivermectin, which is obtained from a fungi known as Streptomyces ivermitilis, which is used as an anti-helminthic agent. And for the production of statins, which are used as an cholesterol lowering agents like lovastatin and pravivastatin from different microorganisms like Aspergillus terras, Penicillium citrinium and Lindius edodes, and for production of clavulinic acid, which is an penicillinase inhibitors, which are used along with the amoxicillin. It is obtained from Streptomyces clavigerus. Now, some immunosuppressants also can be obtained from the fungi like Streptomyces hygroscopicus. It can produce a product like rapamycin, which is used as an immunosuppressant in organ transplant. Then Tulipocladium inflatum, which will give an product cyclosporin. This is another immunosuppressant which is used for the organ transplant. Then fungi can be used for the production of ergotalcaloid, which is an uterocontractant, which is obtained from Claviceps purpurea, and it is used for the induction of labor. And we can produce some anti-tumor agents like doxorubicin, danorubicin from fungi like Streptomyces puscuticus, and mitomycin from Streptomyces pseudosus, and bleomycin from Streptomyces verticulus. All these are used against the cancer treatment. Now, next application of fungus is for the production of antibiotics like penicillin from Penicillium notatum or Penicillium chrysogenum, erythromycin from Streptomyces erythrus, lingomycin from Streptomyces lingolensis, vancomycin from Streptomyces orientalis, streptomycin from Streptomyces griseus, kanamycin from Streptomyces kanamyticus, tobramycin from Streptomyces tenebraceus, neomycin from Streptomyces fradiae, soframycin from Streptomyces decaris, cephalosporin from Cephalosporium acrimonium, and griseofulvin from Penicillium griseofulva. Now coming to the next application for the production of organic acid in industrial scale. Aspergillus nigrus for citric acid production, Rhizopus nigricans for fumaric acid, Aspergillus oryzae for kojic acid, which is an fungicide, and Aspergillus terras for itaconic acid used in production of plastics, Aspergillus niger for gluconic acid. Next application of fungi is production of other compounds in industrial scale. For production of ethanol, we can use Saccharomyces cerevisiae, gibberellin from Gibberellin figicula, insulin, somatostatin, growth hormone, interferon by recombinant DNA technology by using Saccharomyces cerevisiae, and by using steroidal transformation by using different fungus like Rhizopus, we can produce different pharmaceutical ingredients. And soya sauce can be produced from Aspergillus oryzae, and caffeine from Erwinia dissolvens. So that is all about the applications of fungi in pharmaceutical productions. So these are the some questions which are seen from this topic, microbiology of fungi. Write a note on reproduction of fungi. Write a note on applications of fungi. Write a note on industrial applications of microorganism or fungi. Write a note on septate hyphae. Write a note on mycelia. So that is all about the microbiology of fungus. Introduction, classification according to the morphology and taxonomy, cultivation, reproduction and application. Hope it is clear. These are the some references which are used. Thank you for watching this video.